everyone, this is Shalini Tailor, Assistant Professor in Biani Girls College, welcomes you all to my video lecture which is on the topic super coiling of DNA. Let's begin with the definition that what do you mean by super coiling? Coiling, I guess everyone understood that it is the winding of one part or one strand on the another strand, just like the structure of a rope. Okay, it is coiled. But if you intertwine the strand of one strand of rope on the another strand, on the same strand, not another one, on the same strand, then it will form a super coil. That is, super coiling is basically coiling of a coil, right? So when a coiled or the interwound structure is again wound on itself, it forms a supercoiled structure. Basically, supercoiling is being suggested or found in the type of the DNA which are basically circular in nature. And I guess everyone knows that circular DNA is the property of prokaryotes. Prokaryotes do possess circular DNA, but in the eukaryotes, sometimes this supercoiling is also found when the structure of the complete, because as in the eukaryotic organisms, the DNA is very long, and in order to compile it or to form it inside the chromosome, to pack it inside the chromosome, it is being condensed very much, and since due to this condensation supercoiling occurs. Now this supercoiling is of two types basically. It is basically of two types. First one is the positive supercoiling and the second one is the negative supercoiling. Now what is the meaning of positive one and the negative one? Basically, as we all know that the DNA is the double stranded structure. So when this double stranded structure of DNA having the axis on which it is being forming its structure in the form of a helix like this. So this is a double helical structure, and you can see the linear line which is the axis of rotation. So whenever the coiling occurs in a right-handed manner or right-handed fashion, it gives rise to the formation of positive supercoiling, or you can say the clockwise direction of coiling. That is, just suppose or imagine the structure of a rubber band, circular rubber band. When you form a twist in that rubber band, it starts wounding on itself and it creates a number of twists and turns, right? So these twists and turns are responsible for creating the torsional stress. You can say the strain or the stress which is present due to the coiling or supercoiling in the DNA structure. So whenever, suppose this is the double helical structure of DNA, circular DNA, at this state here no type of twist or turn is found in this circular DNA, and you can also call it as a covalently closed circular DNA, that is the triple C DNA. I repeat, covalently closed circular DNA, that is the triple C DNA, which is the generally the structure of the plasmids, right? So when this kind of double stranded circular DNA is present and it does not having any kind of coiling or turns in its structure, it is said to be in a relaxed state. That is, there is no presence of any kind of stress or strain. But if this structure is being twisted or turned like this in the fashion of an 8, then this particular strand of DNA is twisting on the same strand. and here the coiling, or you can say the supercoiling, is being formed, right? Further, if this supercoiling occurs in the manner of a right-handed fashion like this, suppose this is a double-stranded DNA, okay? So this is the right-handed orientation of supercoiling, that is, it will give rise to a positively supercoiled circular DNA. But if the supercoiling occurs in a left-handed manner, that is in the anti-clockwise direction, then it will form like this. This will be the left-handed orientation, or you can say anti-clockwise manner of supercoiling. So there is one important thing or difference between these two, that the positive supercoil requires energy because it creates more amount of tension or the torsional stress when the DNA is being coiled on it again and again. Instead, the type of negative supercoiling, it creates under twisting, means the amount of torsional stress is being reduced when the DNA undergoes negative supercoiling. Why is it so? Because the positive supercoiling creates more amount of condensation of DNA, and the condensation of DNA occurs just because of the twisting and turning. But whenever the DNA is being relaxed or unwounded, like for the process of transcription or replication, or for the involvement of the histone proteins or the freeing of this histone proteins, nucleosomal addition or alterations, then negative supercoiling occurs. Means you just have to remember the thing that whenever the positive supercoiling occurs, then there occurs over twisting or over supercoiling. But whenever the negative supercoiling occurs, it causes under twisting or reduced amount of supercoiling. Clear? Now these method of supercoiling are being depicted, or you can say calculated, using a mathematical expression which is like this.
With world-class education and excellent placement record, Biyani Group of Colleges is committed for your bright career.
Biyani Group of Colleges, where you can trust.
So what is that mathematical expression for the supercoiling? Here three terms are being used, that is the linking number first, second is the twist number, and the third one is the Rayleigh or the width, right? So what is linking number? Linking number means the number of times the two strands are interwound. Just again I am telling that imagine the diagram or the structure of a rubber band, that is the circular one, and when the two strands of the rubber band are interwound on themselves, then it forms a linking number. Say if it is wound in this manner, it is 1. If it is found like this manner, it will give a linking number of 2. Clear? Now what is the twist number, or the twist, simply T, which is represented by T, is the number of times each of the curves rotate around the central axis. As I already told that this is the central axis and this is the structure of a double helical structure of DNA. So when this particular strand crosses this strand completely, then it gives rise to a complete turn, right? A complete round of a DNA strand onto the central axis is the twist number. And the third expression or the third symbol represented by W, which is Rayleigh or the width number, is the number of times the central axis makes loop about itself. Now this particular twist, if again it is in a clockwise manner, will give rise to a positive supercoiling, and if this twist number is a negative integer or in having the negative value, that is in the anti-clockwise direction, then the supercoiling will be negative kind. Clear? So how these how this mathematical expression relates. Whenever this supercoiling in a clockwise direction or in a positive manner, then the twist will be reduced. The twist will be reduced. And if the supercoiling is in the anti-clockwise direction, that is the negative one, then the twist number will be increased to compensate the linking number. Okay. So in this manner, I hope you guys have understood what is supercoiling and how it is being categorized into the positive and the negative one. Now, generally in the prokaryotes, an enzyme named as gyrase is being responsible for releasing this tension of the torsion effect or the supercoiling simply. So this gyrase enzyme is responsible for cleaving or cutting the strand of DNA so that the another strand can pass it off from this one strand and can get into its relaxed manner or mode. And same, if we talk about the eukaryotes, then there are two types of enzyme, topoisomerases first and topoisomerases second are the enzymes which are responsible for releasing these supercoilings in the eukaryotes. This topoisomerase first is ATP independent and it cleaves a single stranded DNA cut, whereas the topoisomerases 2 is ATP dependent and it produces the double stranded DNA cut, right. Whenever the DNA is formed like in this manner, this is the catenated DNA or the linking of a chain, the chain the parts of a chain or the pieces of a chain are interlinked to one another. If we want to release this link, what we have to do, we have to use a topoisomerase enzyme. If we are using topoisomerase type first, then only single strand will be cutted out, and if we are using topoisomerase second, then the complete part of the chain or the double stranded DNA will be cutted out and the strand will be relaxed or released from the another one. So in this manner, two daughter DNA molecules are being separated from one another. It is also present in the prokaryotes. So this particular function of topoisomerases is performed by the gyrase enzyme in case of prokaryotes. Thank you for listening.